Okay everyone, so we just talked in the last video about conjugation, transduction, and transformation, or at least I just introduced these three ways that bacteria can transmit genetic information to other bacterial cells. And before we talk more about conjugation, it's important to understand what is meant by prototrophy and auxotrophy. And again, sorry about my hands. I was doing some plumbing work in my house and they got kind of grimy and having trouble getting it off, the grime off with regular soap. So sorry about that. If you didn't see the last video, I'll have to apologize in each of my videos from today. So prototrophy and auxotrophy, what are these things? Well, before I address that, let's talk a little bit about different types of media that you might grow a microorganism like a bacterium in. So let's assume this is called minimal medium. So it could be different types of minimal medium, but they all have one thing in common. That is, they are made of a simple carbon source, like some sort of sugar like glucose or sucrose, just some sort of carbon source for the microorganism. They have water, they have essential salts, and essential minerals. So prototrophic organisms can grow in or on a minimal medium with just these resources. Prototrophs can make any other biochemical needed for life. So any vitamin, any amino acid, any lipid, the prototroph has all the genetic information necessary to make all those important biochemical compounds, starting with just these materials here. Now, an auxotroph is anything that's not a prototroph. So an auxotroph needs one or more biochemical compounds from its environment in addition to what you would find in a normal minimal medium. So for an example, E. coli, wild type E. coli cells, those are prototrophs. So they can grow on minimal medium. An E. coli, let's say met minus mutant, would require us to add the amino acid methionine to the minimal medium. So requires methionine. Methionine. I think that's right. So requires methionine. So it is auxotrophic for methionine. Again, E. coli wild type prototrophic doesn't require methionine in its medium. An E. coli met minus mutant is mutated in a gene required to make methionine. That means it must get methionine from its environment, must import it in order to survive. So if there's no methionine in the medium, well, this E. coli met minus strain, it will not grow. So also, it's important to understand how scientists work with bacteria like E. coli. They often use Petri plates, little dishes where you can fill them with minimal medium, and you can add agar, which is like a gelatin that can make the medium in a sort of semi-solid, like Jell-O. And that way you can plate bacteria onto the surface of this medium, and the bacteria can grow and use the nutrients from the media. And let's say we took our E. coli wild type strain and we put 20 cells, or let's say 5 cells, because I don't want to draw 10 dots. Let's do 10. 6, 7, 8, 9, 10. Okay, we put 10 cells on this plate of minimal medium, and we do the same thing over here. 10 cells there. So this is minimal medium, MM, and this is minimal medium plus methionine. I have trouble spelling this, methionine. Okay, so we plate the cells out, and they're just single cells, and we spread them out with a little spreader, which kind of can look like that. We spread it around, and we can't see these. Maybe if we had a very powerful microscope and we use some stain or something, we might be able to see them, but we can't see these with the naked eye, so we have to put the plates in an incubator overnight, or you could put them on a bench top, and we'll let the individual cells multiply and form colonies of millions of cells. And when we come in the next day, we'll be able to count them, and we should see 10 colonies. If we put 10 cells on the plate, we should see 10 colonies there the next day. Same thing for this medium over here. Now, if we did the same thing with an E. coli minus strain, a strain of E. coli that requires methionine in the medium, well, put the 10 colonies here, minimal medium plus methionine, and over here is just minimal medium, put the 10 colonies here. Now, these will grow because they can get methionine from the media. They require it. They can't make their own. So they can get the methionine from the medium. They can make all the other compounds they need. And so we'll see our 10 colonies. Well, it might be 11 there, but 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yeah, I put one more. So we'll see our 10 colonies, but over here we won't. So we'll put these 10 colonies down, or 10 cells down, and they won't grow. They won't grow, they won't form colonies because there's no methionine, and they need methionine from the environment. So this is one way we can test different strains of E. coli for different auxotrophies. So let's say we mutate a batch of E. coli and we're checking different cells to see what auxotrophies they have. Well, we can test different media. You know, if they don't grow on minimal medium, but they grow when we add methionine, oh, they must be mutated in a gene to make methionine. Or we can test arginine or lysine or any amino acid or vitamins like biotin. So this is how we can work with bacteria, and we can find if certain strains of bacteria or isolates of bacteria are auxotrophic for certain compounds. So in the next video, we are going to take a look at how Lederberg I'm not really sure how to say his name, and Tatum in the 1940s used auxotrophic mutants to get the first hint that bacteria can transfer genetic information from donor to recipient strains.